Glad to see you here today. Today we're going to hear from Hanna Poffenbarer. Hanna is a candidate for assistant professor of soil nutrient management position. Those of you here to interact with Hanna more during the interview, find her the discussion session today at 2 o'clock in N224 Ag North, and graduate students are invited for lunch. RSVP with Julie for lunch tomorrow in N224 Ag North. So remember those other interactions on your schedule. Hannah's in the midst of a busy time for her, obviously. Next week she's going to be in Tampa at the ASA meeting. November 6th, she's going to be defending her dissertation from Dr. So, Hanna is originally from Maryland, got her bachelor's and MS degrees from the University of Maryland, and also working on her PhD degree at Iowa State. So, Hanna, all yours.
Thank you. Thank you all for being here today. I'm going to talk today about my research in soil fertility, aimed toward improving agricultural productivity, profitability, and environmental performance. So soils are truly the foundation of our agriculture. The soils provide the structural support for plant roots, the pore spaces between mineral particles and aggregates where roots can access water and oxygen, and a huge reservoir of elements essential for plant growth. So one of these nutrients is nitrogen, and just the top foot of soil in a typical Kentucky siltloam soil will hold about 6,000 kilograms of nitrogen per hectare. So it's a huge resource, and most of this nitrogen is actually contained in the soil organic matter, and a portion of this nitrogen is released into mineral forms during the growing season. These are the forms that a plant can take up. So the soil provides many benefits to support plant growth. The plants at the end of the season, they produce biomass, and generally a portion of this material is returned back to the soil as crop residue. And this builds the soil organic matter, a storehouse of nutrients, and it also benefits properties like water holding capacity and soil aggregation. So the soil is a storehouse of many nutrients, but the supply of these elements essential for healthy plants and healthy people, they differ, and they're not always well matched with the quantities that humans need in their bodies. So in the case of nitrogen, we need a lot of nitrogen. We need nitrogen to build proteins, and there's just not a high concentration in the Earth's crust. So we have a very low supply ratio. And humans were on a constant quest for more nitrogen to feed our crops so that we can support more people. The good thing about nitrogen is there's another source of nitrogen besides just what's in the Earth's crust, and that's what's surrounding us in the atmosphere. So about 78% of the atmosphere is dinitrogen gas. Over 1 hectare of land, the atmosphere contains 75 billion kilograms per hectare of nitrogen. So compare that to those 6,000 kilograms in the soil and 100 to 200 kilograms in a crop. We're really swimming in nitrogen, and so this brings us to a paradox. Oh, I also wanted to mention there is a tiny little dot there. I'm not sure. I did actually scale that appropriately to the atmospheric pool, but I'm not sure you can actually see that. So this brings us to our first paradox, our nitrogen paradox, that despite this large atmospheric nitrogen pool, we still see widespread nitrogen limitation. So in almost all of our non-legume crops, we see a response to nitrogen fertilizer inputs. So you can see in this photo, the corn on the left, much larger, darker green, capable of higher yields than the corn that did not receive the nitrogen fertilizer. So humans, and of course the reason for this paradox is that the nitrogen that's in the atmosphere is in a form that's not available for plants. The plants are taking up nitrogen in those mineral forms that are dissolved in the soil solution. So humans have devised ways to transform this dinitrogen gas into the forms that plants can take up. And so these are two major ways that humans add nitrogen to our soil crop systems. We grow legume crops. Legumes are plant species that support microbes in their root nodules. These bacteria can fix atmospheric nitrogen into ammonium, a form that the plants can use. It's an energy-intensive process, so it requires about 94 megajoules of energy per kilogram of nitrogen. And this energy is coming from carbon that was produced through photosynthesis in the plant. So the plant's providing carbon to the microbes, the bacteria in the roots. The bacteria are providing nitrogen for the plant. The other major way that we add nitrogen to, into plant available forms to our cropping systems is the Haber-Bosch nitrogen fixation process. And this is the industrial process where dinitrogen gas reacts with hydrogen gas to form ammonia. It requires intense heat and pressure, a catalyst. It's also very energy intensive. And in this case, the energy is coming from fossil fuels. So to put this into perspective, about 25 gallons of diesel fuel is required to fertilize 1 acre of corn. So these technologies have been really critical in allowing us to increase yields of our cereal crops. This is an example for corn grain in the United States. We could see this, this linear increase since about the 1930s. Our yields have been on the rise. And a number of technologies have contributed. So at the beginning, 1800s, the farmers were growing open pollinated varieties of corn with few external inputs. So maybe manure to replenish nutrients and rotating the corn with legume crops. But the yields were generally stagnant. In the 1930s, Henry Wallace developed hybrid corn. And this was really transformative. All the farmers immediately started planting the hybrid seed. Even though it was more expensive, it resulted in such high yield gains. And so, of course, that hybrid seed company went on to become Pioneer, and Henry Wallace became Secretary of Agriculture and Vice President of the United States. So that was really key technology. And following World War II, we had the availability of synthetic nitrogen fertilizers. So the Haber-Bosch process developed during World War I, and then following the wars, this technology was applied to produce fertilizers. And this really allowed farmers to take full advantage of the genetic potential that came about during this time in the corn. And so this Haber-Bosch process has been hugely transformative, not just for corn, but wheat as well. It's estimated now that average for the world, about a third of the nitrogen in our human bodies is actually Haber-Bosch nitrogen. And for the United States, it's more like 70 or 75%. This technology also allowed not only increased yields, but we could reduce the length of our rotations because we were relying on synthetic nitrogen rather than legumes. We could, in some cases, grow continuous corn rather than alternating with the legume crop. The use of the rates of nitrogen fertilizer application largely stabilized in the 1980s in the US, and we've still seen increasing yields. So this tells us that we're able to achieve higher yields with the same quantity of nitrogen per acre. And increasing plant densities have been a really key factor throughout this history, and especially in recent decades, allowing grain yields to continue to rise. So as we look to the future, we know that the world population continues to grow and place increasing demands on our production systems. And so these technologies and new innovations are going to still continue to be important in the future. Now there's a second paradox, nitrogen paradox, and that is that even though it is limiting in many of our cropping systems, when we add nitrogen fertilizer, it's not used very efficiently. So nitrogen fertilizer is supplementing the mineral nitrogen pool in the soil, that forms of nitrogen that can be taken up by the crop. Also the forms of nitrogen that are easily lost to the environment. So with 100 units of nitrogen applied, usually about 20 to 60% would be taken up by a crop like corn. 20 to 60% taken up by the microbes and then becoming part of the soil organic matter when those microbes produce enzymes and when those microbes die and become part of the organic matter. And then 2 to 15% loss as gaseous emissions, and 2 to 30% loss through nitrate leaching. So these losses are really important because they have environmental consequences. In the case of our gaseous losses, one of these gases is nitrous oxide, a greenhouse gas with over 200 times the global warming potential as carbon dioxide. And agricultural soils are the dominant source of nitrous oxide in this country. So the more nitrogen fertilizer we apply, generally we see higher rates of nitrous oxide emissions. And then we also have impacts on our water bodies. So in this part of the country, agricultural nutrients that leave the cropland and enter rivers, they end up making their way to the Gulf of Mexico. And so nutrients like nitrogen and phosphorus stimulate algal growth in the same way they stimulate crop growth. But then the algae die and decompose. This uses up oxygen in the water, creating a dead zone. So in 2008, Gulf of Mexico Hypoxia Task Force set a goal for 2015. The Gulf dead zone should be less than 5,000 square kilometers. In 2017, this year, the Gulf dead zone is the largest on record, and it's over 4 times the size of that goal that we had set for 2015. So we face a challenge as researchers working in agriculture, and that is to enable economically viable farming systems that can increase productivity and also improve environmental quality. And I think that nutrient management is really central to addressing this challenge. So through my research, I've been investigating controls on carbon and nutrient cycling to improve the precision of nutrient delivery, to enhance soil organic carbon for long-term productivity, and to make more efficient use of our nutrients. So I'm going to talk about research in each of these areas and also provide a little background on myself throughout this talk. So I grew up in Maryland, state located directly adjacent to Chesapeake Bay and very much located at the interface of agriculture and the environment. For many decades, agricultural nutrients within the watershed have contributed to degraded water quality in the bay. And so farmers have really recognized the linkage, especially in Maryland, between the practices on the landscape and water quality. So they've really led the nation in adoption of practices like no-till and cover crops. So I became really interested in soil management to minimize these trade-offs between productivity and environmental quality. I completed my bachelor's and master's degrees at the University of Maryland, focusing in environmental science and soil fertility. And so the topic, or my master's degree research, was really focused on cover crops. Because in our production of summer annual crops like corn and soybean, we have periods of time during the year when the soil and the nutrients within the soil are susceptible to losses. So following harvest of corn in the fall, we may have excess fertilizer nitrogen that's left over at the end of the season, and then is susceptible to leaching if there's no roots to take up the nitrogen. And then in the months leading up to corn growth, we have a period of time where, again, the temperatures may be warming, the soil may be warm and wet and decomposing, forming mineral nitrogen that's susceptible to environmental losses, but there's no plant roots to take it up. And we also may have an earlier season application of fertilizer that's creating this pool of nutrients that can be lost. And so cover crops are one tool that we have to scavenge those nutrients, prevent them from being lost from the system, and also protect the soil from erosion during those sensitive times of the year. So the cover crops are planted in the fall, and they tend to produce some biomass and then go dormant, and then in the springtime accumulate some more growth before termination prior to that cash crop planting. And so after termination, the cover crops start to decompose and release some of those nutrients back to feed the subsequent crop. So one of the goals of my master's research was to use cover crop mixtures to optimize the timing of nitrogen availability, the scavenging, and then the release of those nutrients to the following cash crop. So I combined a cereal rye cover crop. It's a grass that produces good good fall biomass, scavenges nutrients quite well throughout the fall and spring. And then hairy vetch, a legume cover crop, does not produce such great biomass in the fall, so it leaves the soil a bit more bare. But in the springtime, it produces biomass that also fixes nitrogen. It's a legume. And these cover crop species differ also in their residue quality. So cereal rye tends to have a high carbon to nitrogen ratio, hairy vetch a low carbon to nitrogen ratio. This is important because it regulates the decomposition process. With high carbon to nitrogen ratio residue, the microbes are decomposing that organic material. They're using some of the carbon to build microbial biomass. They're mineralizing some of that carbon for energy. But they also need nitrogen to support their microbial biomass. And so if their residue contains a lot of carbon and not a lot of nitrogen, the decomposition process can be slowed down because they become limited by nitrogen. And they take soil mineral nitrogen in the soil solution to fuel their growth, and that reduces the plant available nitrogen supply. In contrast with low carbon to nitrogen ratio residues, the residue provides adequate nitrogen, and so the decomposition process can proceed quite quickly, and in many cases the microbes will actually mineralize nitrogen because they have excess nitrogen. So they release it back into the soil solution. So I hypothesized that with looking at a range of different cover crop species, cover crop mixtures ranging from pure cereal rye to pure hairy vetch, increasing the hairy vetch proportion would lead to more nitrogen content in the cover crop because that legume is providing additional nitrogen, and would also lead to a faster rate of nitrogen release due to the low carbon to nitrogen ratio of that hairy vetch. And this would result in variation in the nitrogen uptake by the corn crop. With pure cereal rye, we'd see inadequate nitrogen supply due to the slow decomposition and the lower carbon nitrogen content of the residue. With pure hairy vetch, we may see suboptimal nitrogen uptake because the residue is decomposing so fast that we have some losses of mineral nitrogen before the corn can take it up. And then a mixture would provide a good supply of nitrogen and also a gradual rate of release that would be well synchronized with corn uptake patterns. So we conducted a study looking at a range of different seeding rates of hairy vetch and cereal rye. This generated different biomass proportions of these two species. And then we measured cover crop nitrogen content as a function of the composition of hairy vetch in the cover crop. So we generally found that increasing hairy vetch led to more nitrogen accumulation in the cover crop. We looked at the proportion of nitrogen in that cover crop coming from the soil scavenging and from biological fixation. So estimated biological fixation using natural abundance of M15. We did find that across this gradient, all of the different cover crops scavenged about the same quantity of soil nitrogen, about 50 kilograms per hectare. And interestingly, in that some of the hairy vetch cereal rye mixtures, we could achieve a similar level of biological fixation as that pure hairy vetch stand. Then we measured the decomposition and nitrogen release of the different cover crop residues. On the y-axis here, you see the nitrogen remaining as a proportion of the initial nitrogen content of the residue. And the x-axis is time. after termination. We have these circles are the pure cereal rye residue, which we can see high C/N ratio. It did not actually release nitrogen. It was mobilizing nitrogen throughout the season. But as we increased the composition of hairy vetch in that cover crop, we found increasing proportion of nitrogen released from the cover crop. And in general, most of the nitrogen was being released in about the first 4 weeks after cover crop termination. So I transformed this into quantities of nitrogen release for 3 of the cover crop treatments. This dotted line is pure cereal rye. We have our mixture, a 50-50 mixture, and pure hairy vetch. And then this green line represents the pattern of corn nitrogen uptake for corn growing in non-nitrogen limiting conditions. So we had a treatment in the field where we applied excess nitrogen just to make sure it was not limiting. And so we can see that actually none of these cover crops provided really the right pattern of nitrogen release. The cover crops are releasing nitrogen most rapidly immediately upon termination. The corn starts taking up nitrogen pretty slowly, and then rapid N uptake occurs a bit later in the season. However, if we compare the cover crop nitrogen release to an application of fertilizer nitrogen at planting, which would be a single pulse of mineral nitrogen to the soil, we can see there's much more potential for losses of mineral nitrogen before the corn is actually taking it up if we're using the fertilizer application. So the next question is, do these cover crops actually supply adequate nitrogen to meet the demands of the corn crop? So this is corn nitrogen uptake in our pure cereal rye mixture, 50-50 mixture, and pure hairy vetch. And this line here represents the nitrogen uptake in that non-nitrogen limiting treatment. And these are 95% confidence intervals. And so in general, the corn N uptake did increase with more vetch in the mixture and the pure vetch stand, but they were less nitrogen uptake than under non-limiting conditions, so we probably were not maximizing our crop growth with these cover crops. But then we added a broadcast poultry litter. We have that as a treatment as well, and you can see that combining either the mixture, the pure hairy vetch, along with poultry litter applied at a rate to meet the phosphorus needs of the crop actually provided a good adequate supply of nitrogen while avoiding excess application of phosphorus. So conclusions from this study, the hairy vetch and hairy vetch cereal rye mixtures provided nitrogen scavenging in the fall and spring, about 50 kilograms per hectare. Improved timing of nitrogen release during the cash crop growth relative to a pre-plant or at planting application of fertilizer, and a sufficient nitrogen supply when combined with poultry litter applied at a phosphorus-based rate. So following my master's degree, I moved to Iowa State University to pursue a PhD right in the center of the Corn Belt, the heart of the Corn Belt, a region of the world. It's actually been identified as the most productive region during the summer months in the whole entire world, even more productive than the tropical rainforest, which I found amazing. This is chlorophyll fluorescence data, and you can see this, and that's a representation of gross primary productivity. And you can see this red splotch here in the Midwest United States. So I came to Iowa to understand these systems, very productive systems, to get a deeper understanding of nutrient cycling and try to apply that understanding to improve nutrient use efficiency. So I already talked about the importance of nitrogen inputs for maximizing productivity within a growing season. It's quite obvious, almost all of the corn acres will see a nitrogen response within that growing season with improved yield. The question is, what is the influence of nitrogen fertilizer application on long-term productivity? How does the nitrogen fertilizer impact soil organic carbon, an important indicator of soil quality an important predictor of crop yield. So there's been a debate in the literature what effect this fertilization has on carbon storage. To understand the debate, it's useful to consider the balance of carbon in the soil. So carbon storage is really a balance of inputs through residue, manure, other organic amendments, and outputs mainly through the decomposition process, but also erosion and leaching losses. So on one hand, with increasing nitrogen fertilizer, we're expected to see higher productivity. This leads to more residue production, more carbon inputs. That should enhance carbon storage, lead to positive carbon storage. On the other hand, the addition of nitrogen may stimulate microbial activity, enhance decomposition through that same process we talked about with the cover crop residues. The nitrogen additions may address a limitation in microbial biomass and enhance decomposition. So this would increase the outputs and lead to a loss of soil organic carbon. So I hypothesized that the impact of nitrogen fertilization on soil organic carbon is probably going to depend on the rate of application and how that relates to crop needs. So this is a typical yield response curve, what you might see for corn response to nitrogen inputs. On the x-axis, you have increasing nitrogen fertilizer inputs. You have this black line representing yield and also residue production, because residue production scales proportionally to crop yield. So with increasing nitrogen inputs, you have increasing yield and residue production, but at some point you reach a plateau where additional nitrogen inputs no longer boost crop yield. And this rate, where the plateau is reached, that's the agronomic optimum nitrogen rate. Above this rate, you may see an accumulation, often see an accumulation of mineral nitrogen in the soil because the fertilizer inputs are supplementing this pool, but the corn is not actually taking it up because it's not needed above that agronomic optimum nitrogen rate. So I hypothesized that because there is a positive relationship between residue inputs and soil organic carbon, that below the agronomic optimum rate, adding nitrogen should lead to increased residue and increased soil organic carbon. But above the agronomic optimum nitrogen rate, we no longer see nitrogen additions improving the soil organic carbon levels. It may plateau just like the residue inputs plateau, or it may actually decline if the accumulation of mineral nitrogen in the soil does indeed enhance the decomposition process. So I tested this hypothesis using 4 long-term nitrogen rate trials in Iowa, and they included continuous corn and corn soybean systems. On the y-axis here is soil organic carbon change. We measured change over 15 years, and then I've annualized it here, so megagrams carbon per hectare per year. The x-axis is fertilizer nitrogen rate scaled as a percent of the agronomic optimum nitrogen rate. So 100% here would be the AONR for each site and each cropping system. So we found that with increasing nitrogen rate, we did see an increase in soil carbon storage. It reached a maximum where we reached that 100% of that optimum nitrogen rate, and then it started to decline. So this was very consistent with the hypothesis that I just proposed. The corn soybean system, however, was much less responsive to nitrogen inputs. We have a intervening legume crop that's not responding to nitrogen inputs, and in general, soybean produces less residue than corn. So we see much diluted effect, and in general, the corn soybean is negative to neutral in terms of carbon storage. Oh, this was 0 to 15 centimeters. So the next question is to determine the legacy impact of nitrogen inputs and corresponding carbon changes on nitrogen use efficiency. Nitrogen use efficiency is an important indicator of environmental performance because it represents the proportion of total nitrogen inputs that are actually recovered by the crop. So increasing efficiency, we have reduced potential for losses. So I used a continuous corn system to address this question because it was a system that provided the most pronounced impact of nitrogen fertilizer on soil organic carbon. So returning for a moment to this yield response curve that I showed, this quadratic plateau response that I'm presenting here is a representation of the law of diminishing returns. The idea that the first input of nitrogen that you apply leads to the largest yield boost. And then as you increase your nitrogen supply, additional units of nitrogen lead to smaller and smaller boosts in crop yield. So this tells us that as we increase nitrogen supply, the nitrogen use efficiency starts to decline. And this is important because we actually do see variation in the nitrogen supply due to the historical nitrogen rate treatments. So this figure shows results from 2 long-term N-rate trials. On the x-axis here is the nitrogen rate that the plots received between 1999 and 2014. And then we measured corn nitrogen uptake in 2015 when we applied 0 nitrogen to all of the plots. So this is corn nitrogen uptake coming with the nitrogen coming just from the soil, not from any fertilizer additions in 2015. So we did find that with increasing historical nitrogen rate, the nitrogen supply and capacity of the soil increased. The corn was able to take up more nitrogen in plots that had previously received large inputs. So this leads us to the hypothesis that increasing historical nitrogen rate and increasing soil organic carbon levels should reduce the nitrogen use efficiency because we're increasing the nitrogen supply, and as we increase nitrogen supply, nitrogen use efficiency goes down. But there's another law that we need to consider, and that's the law of optimum. And the law of optimum is that a resource that's in limiting supply will be used more efficiently when all the other resources are optimally supplied. An example would be looking at the nitrogen response curve in a low quality soil and a high quality soil. You would expect to see greater yields even at the same nitrogen rate in corn growing in a soil with very good water holding capacity, good soil structure, good supply of other nutrients. So a high quality soil, you'll be able to achieve a higher nitrogen use efficiency. And in general, soil organic carbon is a good indicator of soil quality. So this leads to the hypothesis that, the contrasting hypothesis, that improving or increasing historical nitrogen rate improves conditions for crop growth by enhancing the soil organic matter, and that should lead to higher nitrogen use efficiency. So testing these hypotheses, we set up subplots within these long-term nitrogen rate trials at 2 locations. The 0 nitrogen subplot, you saw some of the data I just presented a few slides ago from those subplots. And then we also applied nitrogen at a constant optimal rate across all these plots that had historically received different rates. And we used N15 labeled fertilizer so we could trace the fertilizer recovery into the crop, into fast and slow soil organic matter turnover, soil organic matter pools, and by difference we could determine losses of nitrogen as well. So in this figure we see fertilizer recovery as a percentage of the total fertilizer applied at that optimum rate, a measure of nitrogen use efficiency. On the x-axis, we see the historical nitrogen rate between 1999 and 2014. And we can see that there were dramatic differences between these 2 locations in nitrogen use efficiency. Central Iowa was able to recover about between 40 and 50% of the fertilizer nitrogen. Southern Iowa was really terrible, really terrible nitrogen use efficiency, down between 10 and 20%. But the amazing thing actually was that they both showed this similar curvilinear response. It was pretty subtle, but it was a statistically significant quadratic curve there. And it does suggest that there may be two processes controlling this response. So it aligns with the idea that we may see a decrease in nitrogen use efficiency due to changes in the nitrogen supply. We may see an increase in nitrogen use efficiency due to improved soil organic carbon and soil quality. So we're looking at the nitrogen recovery, fertilizer recovery in the soil organic matter at different times during the season and the total soil, bulk soil, to get a better handle on the mechanisms that underlie this response. So to conclude, the long-term nitrogen fertilization was important for maintaining or increasing soil organic carbon in the dominant cropping systems of the Corn Belt, and that it does appear to impact nitrogen use efficiency, possibly by affecting the nitrogen supplying capacity of the soil and the soil quality. So I've talked today about 3 research areas that I focused on: investigating cover crop mixtures to improve timing of nutrient release, nitrogen fertilization and crop rotations to control soil organic carbon storage, and nitrogen fertilization practices and their impact on nitrogen use efficiency. Looking into the future, coming to the University of Kentucky, if I have this opportunity, I will continue to work in these areas and I'll present a couple of research ideas to finish up here. So within the realm of precise nutrient delivery, I'm interested in investigating the factors that control spatial and temporal variation in nutrient supply. I'm looking within the root zone, interactions between the soil microbes, the organic matter, plant roots, and mineral nutrients, how these interact to affect spatial distribution and proximity of nutrients to the roots, as well as the fluxes of nutrients throughout the growing season. Also interested in investigating this question at a subfield to field scale and among different years. I'm looking at factors like soil properties, landscape position, weather patterns, how they interact with crop residue and management to affect nutrient transformations, and then using this information to identify where, what conditions we should modify, timing, rate, placement, source to provide adequate nutrients to the crops while avoiding environmental losses. Within the realm of long-term productivity, I'm interested in investigating factors that control soil organic carbon stabilization and mineralization, factors like tillage and crop residue quality, root distribution, how these may impact storage of carbon within slow turnover pools like mineral-associated organic matter and aggregate occluded fractions, as well as the faster turnover pools like particulate organic matter, with the goal of achieving both carbon storage for long-term soil quality and maintaining mineralization processes that provide these fluxes of nitrogen, phosphorus, and sulfur that are helpful for plant growth. And finally, applying the basic understanding of carbon and nutrient cycling at a systems level to enhance, identify ways to enhance resource use efficiency with the goal, and really evaluating these systems and using multiple indicators with the goal of developing systems that are going to be more productive, more profitable, and better for the environment for Kentucky and beyond. So I wanted to acknowledge the individuals at University of Maryland and Iowa State funding agencies that have supported this work, and thank you all for your attention. I'd be happy to take your questions. Yes. Mm-hmm. Yes. Those long-term nitrogen rate trials, they all received chisel tillage in the fall. and then secondary tillage in the spring. And the nitrogen source is either urea incorporated in the spring or UAN applied as side dress application. Yes. That's a good question. So, yeah, the thing about the southern site is that it has a really high agronomic optimum nitrogen rate. So it's one of these sites where in many years, even at the highest nitrogen rate that's applied in those experiments, you'll still see an increasing yield. So it doesn't actually plateau. So it requires a ton of nitrogen to maximize the yield, but the yields are really low. So it's really, I would identify it as a location where it's not good for growing corn because you have to apply a lot of inputs and you don't get a lot of yield. So I think this combination of high agronomic optimum nitrogen rate, low yield is due to just the potential for losses. It's very poorly drained and it doesn't have tile drainage. My guess is that a lot of it is lost through denitrification in the springtime. Yes, right. Mm-hmm. Right. Mm-hmm. Yes. Mm-hmm. Yeah, the tillage. Okay, well, I guess I'll start with the first part. So yeah, in the case of a legume crop like soybean, I don't think we would see a response, just because the soybean is typically not responsive to nitrogen inputs, and that's why I think we saw with the let's see, if I can go to it. With the corn soybean rotation, such a less pronounced response because we don't have increased residue production coming from nitrogen additions in 50% of the year. So I think the most accentuated impact of nitrogen fertilization on soil organic carbon would be in a really productive C4 crop like corn or maybe like sorghum. That is really responsive to nitrogen fertilizer and produces a lot of biomass. But I think in most non-legume crops, you probably see somewhat of this effect. It may depend really on the previous history of that site. So one of the reasons I think we saw the potential for an increase with well-fertilized continuous corn is that previously these plots had been in corn soybean rotation. And so we've switched to a system that is adding more carbon year in and year out. So we're able to actually boost the carbon. But if you're in a system that was in corn soybean and then you continued in corn soybean, there's pretty much a neutral impact. For the tillage, I think that would be an interesting thing to look at because we know that no-till can definitely increase organic carbon in the surface layers, but when we look at the whole profile, appears to be mainly just a redistribution of carbon, that we can actually see the same quantities of carbon if we look deeper down. So there could be interesting mechanisms. The proximity of the residue to the soil particles may enhance the storage of the organic matter in those mineral associated fractions. So I think that would be an interesting area to explore. Yes. Okay. Yeah, so one of the things, I may even actually have this as an extra slide. During my master's degree, I got interested in this idea, and I did some work with spatial statistics to do, this is actually just within the root zone, the different depth and then distance on the x-axis from the corn interrow center. So techniques like this that would allow us to actually quantify the spatial distribution, and this could either be within the root zone or within a field. I'm really interested in modeling. In both my work at Maryland and at Iowa State, I've worked with modelers to include my results to try to improve the models. So at the University of Maryland, I worked with a modeler at Cornell to improve the precision nitrogen management model to be able to simulate cover crop decomposition. And so to me, that's really a key next step to be able to bring the results into improving the model, a place where we can formalize the state of the science, understand the gaps that exist in our understanding, use that model to evaluate a wider range of scenarios. So I think a model would be really helpful in understanding the year-to-year variation in nitrogen needs, because we can evaluate a wide variety of weather conditions that you wouldn't be able to capture in a field experiment. Mm-hmm.
And I could just ask if anyone else.
Oh, does anybody at Princeton have a question?
Okay. Well, I'll have a question for you about the biological ways of matching nitrogen to crop uptake. And added poultry to this one time, or are there physical or chemical ways to match nitrogen?
There.
Yeah, so one thing that comes to mind are the specialized fertilizer products that, so they have like urease inhibitors or nitrification inhibitors that can slow down some of those nitrogen transformations to reduce the risk of losses. So in Iowa, a lot of farmers will use N-Serve to prevent nitrification after they apply anhydrous ammonia in the fall. So that's one example of a chemical method to try to better match the nitrogen supply with the uptake by the crop. Physical methods, I'm really excited actually about, and I kind of showed it with this one picture, this idea of soil functional zone management, where things like strip tillage or ridge tillage, where you're managing the soil, physically managing the soil to create a zone where there's faster decomposition, so there's a pulse of nutrients right in that where that crop row would be, and then you also have a zone where you're kind of storing carbon. You have physical protection of the soil. You have the ability to suppress weeds. You have different zones that are providing different functions, and this can help manage the fluxes over time and space through physical means. I don't know if you have something else in mind with physical. Okay. Mm-hmm. So we prepared the samples, put them in little capsules, but then we ended up sending them out for isotope ratio mass spectrometry. We've been sending them to Utah State Stable Isotope Lab. Mm-hmm.
Okay, so just to remind you, we'll have a discussion session with Hanna at 2 o'clock today in 224 Ag North. So, thanks, Hanna, for our presentation.